Hi, this is Mike Healy. This video clip is part of a long-distance training course in how to diagnose turfgrass diseases by microscopic examination and is geared for or to the turfgrass manager. This course is a result that in many areas of the world do not have access to a convenient turfgrass disease diagnostics lab, and also the ever-increasing difficulty of sending samples outside one's country. This course requires that the turfgrass manager taking the course has on hand and has learned how to use several different types of microscopes along with an ability to capture digital images viewed in both microscopes. Virtually all communication between instructor and student will be through the internet. An ability to communicate via Skype will also be very important. As much of this course content will be by video clip, ability to access YouTube will also be very important. This case study concept about to be outlined with case study 01 example will be an integral part of course content. This slide is pretty much self-explanatory as to what the purpose of case studies are and what will be presented. All case studies to be created are based on diagnoses already carried out in my laboratory, literally some 1,000s in the last 10 years. I'll be selecting case studies I believe have the greatest training value. This slide gives you the background of the stand of turf to be diagnosed. In the geographic area where the problem is located, almost always such stands of turf are overseeded. This year, this particular stand of Tifway 419 Bermuda grass turf was not overseeded. Mild weather kept this grass growing actively even into early February. As you will see in the next slide, the field symptom photo provided by the turf manager, the primary symptom is leaf yellowing. The initial thought on the part of the turf manager was that the problem may be nutritional in nature. You can see patches of yellow turf in this photo. One cup cutter plug of turf, taken half in and half out of a yellow area, was supplied by the turf manager to be examined for possible disease. He had already just taken soil samples for soil fertility analysis. When the sample was received by my laboratory, it was thoroughly washed and then placed in a plastic container with wet paper toweling and sealed so as to create a 100% relative humidity environment, kept at room temperature, and examined beginning 12 hours after the beginning of incubation. The first observation I made was to examine rhizomes with a dissecting scope, resulting in finding dark fungal mycelium growing on the surface of many rhizomes. This photograph was taken from the dissecting scope. A sliver of rhizome sheath tissue containing the dark mycelium was transferred to a microscope slide, bathed in water, covered with a cover slip, and then examined using my standard light microscope. This close-up view of the mycelium found numerous right-angle branching of mycelium normally associated with the Rhizoctonia fungus. If there had been special infection structures called hyphopodia, then this would have been used to identify the sample not as of Rhizoctonia, but as one of the decline fungi, most likely the Bermuda grass decline found in our area. At plus 36 hours post-incubation, fungal growth was found in many leaves. The picture you see is one taken through the dissecting scope. At the upper right area, there is visible both fungal spore production structures with attached fungal stores. One of these leaves was removed with the tweezers, placed on a microscope slide, bathed in water, covered with a cover slip, and examined using a standard light microscope. Spores produced by this actively growing fungus are most likely those of Drosera, formerly called Helminthosporium. Here in the southeast USA, I almost always find this fungus as a disease in winter and often causing yellowing of turf. Take a second look at the turf field symptom photo. Can you see what appears to be a more or less straight line of yellowing beginning in the lower center of the photo? The turf manager also believed this specific inline yellowing might be due to a post-emergent herbicide application overlap problem. The herbicide had been recently applied. The problem is that in many areas of yellowing, there is no noticeable inline symptom observed. As we conclude this case study, the cause of yellowing has not yet been conclusively identified. The turf manager, in advance of soil test results, has made several test strips of various nutrients and applied a test strip of chlorthalonol to determine whether or not Dresleuria leaf disease was the primary problem. And if neither one of these treatment strategies proves effective, then do we blame the herbicide burn via spray overlap? The time taken by the turf manager to rule out various causes will be valuable information for him to have in future years.